Dear students, we can have a discussion on the bioreactor. You might have heard about the nuclear reactor in which nuclear reaction takes place to produce nuclear energy. But in bioreactor, microorganisms act on so many nutrients and bring about the production of some products at industry level. In order to produce desirable products at industry level, large culture vessels are used in which microorganisms are cultured at a consistent level or in a consistent manner, and microbial products are produced continuously and these are marketed. This picture shows the diagrammatic representation of a bioreactor, where you can see the large vessel has got many layers. The outer layer has got, see here, it's the outer layer. Outer layer is a covering in which coolant water has been filled. Coolant water has been filled. Through this water, cooling water is pumped into the chamber, and through this vessel, cooling water will be pumped out. We can see the components of bioreactor in details. Here you can see motorized stirrers. Motorized stirrers are used for the mixing up of the microorganisms with the nutrients. For the continuous and consistent mixing, this motorized stirrer with the paddles below are utilized. Here you can see, for achieving a constant motion, and it will ensure the agitation between the nutrients and the microorganisms. Then another part is the baffles. Here you can see the baffle, this one. These baffles are flat plates which are used to prevent the adsorption of microbes and substances to the surfaces of the bioreactor, and it also ensures the distribution of air, oxygen and other gases in the bioreactor. So this baffle will be rotating through the walls of the bioreactor inside, and it will prevent the adsorption of the microorganisms and the substances on the walls of the bioreactor, and it will also ensure the distribution of air, oxygen and other gases in the bioreactor. Then aerator system for letting in sterile air or other gases evenly and without injuring the cells. See here, sterilized steam inlet, sterilizing steam inlet through which sterilized air is pumped in, and it will provide the supply of oxygen and other essential gases. The next one, foam breakers. Foam means the production of foam will be there during the production or during the reaction of the microbes with the substances. So this production of foam will be prevented, and the flocculent formation will also be prevented. What is meant by flocculation? Flocculation means the aggregation of substances during the continuous agitations. Just like as you have seen in the case of sewage treatment, there will be aggregation of suspended particles and they will be settled. But if it happens during this bioreaction, it will be negative, because there should not be the aggregate formation and settling. Instead, there should be consistent and uniform mixing of all solute and solvent here. So in order to prevent the foam formation and flocculent formation, there will be a foam breaker, that is here. Next one, thermostat. Thermostat is an instrument used to monitor and maintain temperature at a constant temperature. Then cooling and heating jackets to maintain proper temperature conditions. Then biosensors will monitor the pH, level of nutrients, gases such as oxygen and carbon dioxide, then presence of toxic substances. These will be sensed by the biosensors, and it will be useful for getting information about the condition of the bioreactor inside. Then chemostat will be used to get the information on the level of the pH, buffer salt, chemicals and other gases inside. Here you can see thermostat, then pH control reservoir. Next part is sterilization systems to provide the convenience of in situ sterilization. In order to bring about the sterilization of the products and the whole reaction, there will be a sterilization system and the safety valves for the protection against unexpected increase in pressure that you have seen in the case of pressure cooker. There will be a safety valve. Here it is. Then these are the components of the bioreactor. Now we can see the processes involved in the bioreactor technology. If we have to produce a particular product at industrial level in large scale, we have to first select the suitable microorganism for the same, and then we have to decide which type of reactor will be suitable for the purpose, and then the nutrients essential for the microbial or microorganisms have to be selected. Then this setup will be done in a small scale at laboratory level, and the total process will be standardized, and the fitness and the efficiency of the production will be tested in the laboratory. Then it will be implemented at industrial level in large scale. And in the bioreaction, all these reactions will take place and suitable or desirable product will be obtained. Here you can see other parts also which are not mentioned in the textbook. You can see here nutrient inlet, through which essential nutrients for the microorganism to act upon will be delivered to the bioreactor. Then microbe inoculation inlet. Through this inlet, microorganisms are continuously supplied to the bioreactor. Then filtered air inlet for the aerobic microbes. Aerobic microorganisms need continuous supply of oxygen, and the filtered air will be passed to the bioreactor through this inlet. Now we can see the advantages of using microbes at industrial level. There is nothing to explain here, but I'll just read out. The raw materials used are renewable organic materials, often those which are considered as waste generated from other uses. During the formation of sugar, a lot of byproducts like molasses or the wastage of sugar cane, these will be utilized as a raw material for the action of yeast and other microorganisms, and these molasses are used in large scale at so many industries. These are only an example. Then reduces the dependency on fossil fuels. Then if these microorganisms, since these are microorganisms which act on the nutrients and bring about the production, there is no need of continuous supply of electricity or petroleum fuels for the reaction. Instead, the microbial organism will produce these products as a result of their metabolism itself. Then production of carbon dioxide, the greenhouse gas. Then these pollutant gases are not produced, or these are nil or drastically low. Then drastically reduces or produces no pollution, again the same point. Then products manufactured can be recycled. The products manufactured in this bioreactor can be recycled and used for versatile purposes. The reduced dependency on electricity, which have been already discussed. Then byproducts can be utilized as fertilizers or biofuels. During the reaction of microorganisms, along with the desirable products, there will be some byproducts which can be utilized as fertilizers or as biofuels. A lot of examples will be there. Since the waste produced are organic and recyclable, the burden of waste disposal is minimized. That's a very good point, because in every industrial setup there will be some effluent and waste. They have to be discarded without any harm to the environment. But since the waste produced in bioreactor are organic and recyclable, the microorganisms in the environment will act upon and degrade that. So the burden of waste disposal or waste management is comparably nil. Then sustainability of the process in each batch and in any location. If we combine all these points, we can see that the total usage of bioreactor is sustainable because it doesn't utilize any environmental resources without any reason, and it produces no pollution. Then last point, reduced cost of production. That will be understood if we have seen the way of production of the microbial products, because these microbial organisms produce these desirable products as a result of their metabolism, and there is no any synthetic or artificial chemical reaction involved.